Bismillahirrahmanirrahim. My name is Mubasher Khan, Department of Microbiology, MPhil second semester. Our course instructor are Dr. Niaz Muhammad, Associate Professor, KHAN University of Science and Technology. Topic are effect of arbuscular mycorrhizal fungi on growth parameter of Pezium sativum. Their impact factor are 3.21. Presentation outline or research article, introduction, aim of the research, material and method, result, conclusion and references. These are a research article that I have taken from Asian Journal of Mycology. Introduction. Pea are commonly grown leguminous crop and its edible seed. Pea is placed among one of the oldest cultivated crop. Pea seed are good source of vitamin A, B, C, E, zinc, cholesterol and other antioxidants. About 90% of the terrestrial plants are colonized by mycorrhizal fungi. Mycorrhizal fungi are associated with the plant and establish mutualistic symbiotic relationship with fine plant roots. These fungi help in enhancing plant health to combat both biotic and abiotic stresses. These fungi grow in plant rhizosphere and help in the increased absorption of immobile fixed iron, water and nitrogen. The fungus benefited by receiving carbon compound provided by the host plant. The effect of mycorrhizal fungi were examined in terms of plant length, leaf length, total number of leaves and root length. The spots were filled with a mixture of sterilized soil and sterilized farmyard manure. The labeled experimental spots were shown with 2 pea seeds in each. These spots were inoculated by using 4 mycorrhizal fungi, namely Glomerulus intradesi, Glomerulus aggregatum, Glomerulus sclerum and sclerucided microcarpa. The aim of the research was the effect of mycorrhizal fungi on some of the growth parameter of a pea plant. Material and method. Soil was collected from the experimental field of the Abelish University to be used. The spots experiment and sterilized by autoclaving, followed by air drying to remove the excess of moisture. Sterilized farmyard manure was also added in air dried soil in the ratio of 3:1 and mixture was then filled with the labeled spots. The 2 seeds were placed in each spot at the depth of 2 to 3 cm. The total of 9 spots of each treatment and control were maintained for every experiment. Isolation of the fungi. 25 grams of rhizospheric soil of pea plant was taken and added to the 100 ml of water for uniform suspension. The suspension was then passed through the different size of sieve and finally through Whatman filter paper. Filter paper was examined for spores. The spores were transferred to a glass slide for identification. Microscopic characteristic of mycorrhizal fungi. The identification of mycorrhizal fungi was carried out based on diameter, wall-wide, colors present and absence of hyphae. The microscopic observation revealed that the spores of the intradesi were dark black with hyphae. The spores of aggregatum were dark brown to yellow without hyphae. The light brown spores were observed in case of sclerum. The dark brown to yellow spores of micro. In figure A show intradesi, in B show aggregatum, in picture C show sclerum and picture D show microcarpa. Result. The result of the present study were expressed in terms of microscopic characteristics of 4 mycorrhizal fungi used. Different morphological characteristics was plant height, leaf length and total number of root leaves and root length of the pea plant were examined. It was observed that these fungi have significant effect on plant height, root and number of leaves. They do not have any significant effect of the root length and number of leaves. These are the table that we have separate in graph. The table 2 show effect of glomerulus and tritis on different characteristics of the plant. They can check that treated plant separate and control graph have separated. The the two dots the two dots show that have a significant effect of the growth, and three dots have highly significant. In table 3 they show have that glomerulus aggregatum. The table 4 show that glomerulus sclerum on different characteristics of pea plant, and table 5 show that effect of sclerocyted microcarpa on different characteristics of pea plant. Conclusion: all the four fungi have significant effect on the different parameter of pea plant. The intradices show have best result on the plant height and aggregatum on the leaf length. The sclerum had best result on number of leaves in root length and total number of leaves. The sclerum and microcarpa show have no significant result. These are the references that I have taken. Thank you for your precious time. If any confusion regarding the topic, feel free to ask in comment section.